All right, hopefully everyone can see my screen okay. And let's go into slideshow mode here. All right, so really excited to talk to you about a couple of these quick tools today. So 2 topics we'll cover. First, collecting data from SRA, the sequence read archive, and subsetting or downsampling that data on AnVIL. So please feel free to use any of these slides. It's nice if you credit us, of course, but we're always down for more sharing of education. So feel free to spread the word. All right, we'll talk a little bit about some background of just why you might want to do these 2 actions with your data or use them on AnVIL. And then we'll actually get into the fun stuff, the demo of a workflow called SRA Fetch from DocStore, and then a demo of a workflow that we put together as a team called FastQ Subsample. All right, we'll get into the background first. All right, so by now, many of us working adjacent to genomics or genetics have seen this particular kind of piece of data where the cost to sequence a genome has just gone down precipitously, even in the past 10 years or so. We've seen really amazing breakthroughs and are now able to sequence genomes for about $1,000. So cost is really not as much of a limiter to collect genomic data as it has been in the past. But this does create other problems. So our worldwide capacity to sequence genomes is about 2 million human genomes a year. So that's a ton of data. We're on track to have 1 exabase pair per year by 2025. So that's just around the corner. And on track to create a zetabase pair of data per year by 2035. You may be asking what these increments are, but let's talk about that real quick. And so I always love this table. You know, we're used to thinking about mega bytes and gigabytes and things like that, but we can also think about that in terms of base pairs as well. So for reference, human genome is about 3 gigabases, and we're dealing with zetabases by just a few decades, or almost a decade and a half from now. And just to put this in some other terms, if we think about how much storage a phone has, usually around 64 gigabytes, we would need about a phone and a half to store a genome. So if we think about having 100 zettabytes of data, that's 15.6 billion iPhones. And we could go to the moon 5.5 times, or New York City to LA 569 times if we were to stack these phones up. So this is just a ton of data, and it's currently exponentially growing. It's not just in genomic formats, but in all these really complicated, complex, unstructured data formats like text, video, images, all that kind of good stuff. And so all that being said, we have so many sources of free data and databases online. So just genomic data alone, we have the amazing resources at GenBank. We have dbGaP, databases of SNPs for polymorphisms among samples, you name it. There's so much data already out there. And one of these sources of data that you may encounter or may be looking for data at is the sequence read archive. And just by their own definition, the SRA is the largest publicly available repository of high throughput sequencing data. So it's got data from all branches of life, metagenomic data, environmental data, and individual organism data. So just tons of resources here. And suffice it to say, if you were to kind of search a topic that was of interest to you, you get a ton of results. So not just data sets, but individual samples. Basically, there's unlimited data. And so instead of creating new data, I think there is just a wealth of opportunities to do secondary analysis out there. And if the data already exists in this archive, why not do some follow-up analysis on this data, or kind of a meta-analytical approach using data that already exists. And the nice thing is this is actually pretty easy to bring into platforms like AnVIL. You never have to download the data onto your computer to start working with it, because that process can be pretty slow. Shifting gears a little bit, let's talk a little bit about subsampling. Also, we'll call this downsampling as well. This process is really useful for situations where original data is very large. So that definitely applies to genomic data and genetic data. And this process helps you reduce the size, either for computational efficiency or to facilitate some kind of quick analysis. If you're like me and your code is a little duct taped together, sometimes it can be useful for debugging too, right? You're not throwing the largest file you have at your code right away, but just making sure things run appropriately before you dive in. And so there's lots of ways to do downsampling or subsampling. You can do it in kind of a way that you sample strategically from different groups. Like in this example, we've sampled just a small proportion of each of the different color groups. But for genomic sequencing data, we could take a naive approach too. We could just say, we have hundreds of thousands of reads in the sample, but maybe I just want to start with a handful of reads to make sure my code will run and the analysis works properly before I send the whole data set. Okay, so with that background, let's get into one of the workflows that can help us bring SRA data onto AnVIL, again, without having to touch or download that file to our local machine or to a server or anything like that. All right, so let's go. Okay, so I have a workspace set up here, just called SRA data on AnVIL. There's really not much going on in this workspace. There's not even a description, but I do have in my data tab a set of samples. Okay, and this is really just bare bones. It's SRA IDs. So you can see I've got 5 IDs here, nothing too complicated. I'm going to go ahead and clone this workspace just to show that these SRA IDs and actually the workflows as well will be carried over into the next workspace. So I've got here in my workflows a workflow called SRA fetch, and we'll talk more about that in just a moment. So let's go over here to the 3-dot button here, and we'll clone the workspace. I'll just call this demo. This will take a moment. All the magic happening on the back end. And while that's happening, I just have the SRA open over here. Again, you can search any of those terms you're interested in. Say I'm interested in doing some soil sequencing. I can see there's over 1 million results here. So lots of work has been done in this space. Okay, so my workspace has been cloned. I still have in this sample table those sequence read archive IDs, and I still have that SRA fetch workflow. So anytime you clone a workspace, those workflows are going to be carried over as well. So let's dive into this SRA fetch workflow a little bit. Okay, so the nice thing is I think AnVIL has this really great capability built in where you can see the source of the code that's actually going to run on GitHub. And so you can kind of see every step of the way what's actually happening. If you want to kind of change things or build your own workflow, you can start with some resources that already exist. You don't have to reinvent the wheel from scratch. But let's go ahead and run this. We'll want to use this version. That's all good. And we'll want to run the workflow with inputs defined by a data table, because remember we have those IDs over in that data tab. Okay, I'll go ahead and select sample and select the data. And I feel pretty good that this workflow is going to run correctly. I'm going to just go ahead and select all the samples with this top checkbox and hit OK. All right. Taking it down here to the inputs and outputs, I only have one attribute that's required. And this is, it tells you here, it wants to know what the SRA accession number is, or that ID that we looked at in the table earlier. I only have one column in my data, so it's pretty straightforward to select which one that is for the input. For the output, you can see here that I'm expecting to see a forward read and a reverse read to basically two paired end reads. And it's going to output columns read 1 and read 2 in my sample data, assuming it runs correctly. Okay, I'll go ahead and save these inputs. And I'll go ahead and run it. And I feel pretty good about this. We're going to go ahead and run it even. I don't know how much it's going to cost, but I'm going to go ahead and run it. We'll talk more about that in a moment. All right. So things are queued up. This will take just a minute or two. So let's talk a little bit about some of the data that we're working with. Let's go back to this data tab and my samples. I've got, again, my sample IDs here, but that doesn't really tell us much about the experiment. I'm going to go ahead and copy that ID, and I'm going to look it up. I'm just kind of curious what's going on with this sample. Okay, so I can get a little bit of information about where this data was collected. It's from Northern Illinois University, and it's actually soil metagenome data. So we've been thinking a lot about this in our genomic data science community network about how this is a really cool way to get started with genomic data. This particular soil sample was collected from tallgrass prairie, which is near and dear to my heart. It's I did my grad work on. So a little bit more info. If I click the link here, I can see it's 16S sequences. So it's not whole genome or shotgun information, but rather a limited set of DNA. And I can, again, see more information about that if I like. Important too to note that this is a single end. So actually, we're not going to get that reverse read that we might expect for some samples. All right, let's go ahead and check on our jobs. Still submitted, still running, but it's been submitted, so no longer in the queue. Again, I can see all of these are running in parallel, right? So one sample doesn't have to finish before I run the next one to bring that into my workspace, which would contrast it with how we do it manually. If we were downloading those files onto our computer, right, we're going to have to do really one at a time. And it's pretty difficult to parallelize that download process if we're working on our local machine. Okay. Any questions up to this point? We'll have just another minute for this to finish up. But I want to pause for just a moment.
Ava, do you know how long this process usually takes? I mean, does it depend on, I'm assuming it depends on how many samples you're pulling, but does it depend on anything else?
Yeah, that's a great question. So in my experience, at least with this particular workflow, it does parallelize things. So it really doesn't matter if we're submitting one sample or we're submitting 10. It's able to kind of structure that across the compute. But in my experience, this takes like maybe 2 or 3 minutes. And most of that is just kind of orchestrating the resources, making sure that gets done appropriately. And the running itself of bringing the data into your workspace and moving it into your bucket actually doesn't take too long. Yeah, but I might be eating my words a little bit. It might be taking a little longer than usual. Um,
I also have a question.
yeah, yeah.
So this workflow is pulling data from SRA. Um, would it
Yes.
be possible to make a workflow to pull data from other databases or modify this workflow?
Yeah, so like I said, the source code for this workflow is right here. So if you wanted to make those changes to refer to a specific workflow, or sorry, a specific database, you can kind of create your own workflows in a pretty straightforward way. But the cool thing is, like, I didn't create this workflow. This was available on DocStore. If you see the source here, you can find workflows that do a lot of different things. And so I haven't explored this too much, but I imagine if you went to DocStore and you were interested in a specific database, you could search to see if a workflow already exists that does that process. All right. Well, fantastic. The workflow finished. Thanks for the questions to kind of keep us busy there. If I go ahead and click on this, I can see that the samples were all brought in successfully, which is pretty cool. I won't get a run cost until I believe 24 hours later, but just to give you an idea, when I've run this before, the cost is it's literally less than 1 cent. And so we're dealing with micro cents here. So you'd have to bring in quite a few samples to get up into the dollar range. All right, let's take a look at the data. I can now see I have the sample IDs, but now, like I said, I have those 2 new columns. It was only single end reads, so I have the FASTQ gunzipped files here. These are linked out to actual files in a bucket. And I don't have a read to because it's just single end. All right. Going back to our slides here. I did want to point out that all of the information that I'm going to talk about right now is all in a self-guided tutorial. So you can find the link here. And let me actually drop the link to the slides in the chat. Because I want us all to have access to that. And we'll put those in the YouTube as well, description. But yeah, all this information is online. If you're like me, it kind of can be hard to follow something live without kind of scrutinizing and thinking about it. So if you ever want to revisit any of this, it's all in this tutorial here. All right, so let's talk a little bit about the FASTQ subsample workflow on AnVIL. So let's go back to our workspace. And we're actually going to bring this workflow in from scratch. It's not already here, so we're going to have to find a workflow, and we're going to have to go check out DocStore. And like I was saying earlier, you can search for workflows to do any number of things on AnVIL, whether it's accessing with another database. You can always check to see if those exist already, because that's easier than writing your own from scratch. So let's check out FASTQ subsample. Boom, our workflow is here. You can check it out if you want to get more information and look at the code. That's all set up on GitHub, and you can check that out there. And the nice thing is it's pretty straightforward to export these back to AnVIL. I'll go ahead and click there. I'm okay keeping the workflow name the same, but I want to do SRA demo and import it. You could also create a new workspace if you wanted. Okay. So I've got the workflow already here. Again, the code is right there if you want to check it out. And I'm going to need to define the inputs and data just the same as before. Okay. Again, I want to run it on all the samples. I feel pretty good about that. Let's go ahead and click OK. And this time the inputs are a little bit more complicated, right? There's not just one field that I have to refer to. So let's take a look down here. Our inputs say, okay, it's running in this kind of main workflow. I want to have a variable for the first read of read file, and I want to provide that here. This time I have 3 columns in my data. I don't want the sample ID. That's just the SRA identification number. I want to provide that first read. Okay. And then here I want to provide the sample ID, just the name of the sample. Here I have the option to provide a second read. We don't have one, so I'm just going to leave that blank. And then for this final attribute, you can get more information on what these actually are, again, on DocStore and on the documentation for the workflow. But this is basically how many reads I want to take from that overall sample. And let's say I want to take just 1,000 reads, just a random subset there. And I feel pretty good about this. Let's go ahead and look at output. And again, this is pretty straightforward. It's going to create a file that's a subsample of the first read and of the second read if it exists. Okay, let's go ahead and save. And we're ready to run. Let's go ahead and launch. Okay, again, status here is submitted. If I go to my job history, I can see that the number of workflows, it's running 5 in parallel for all 5 of my samples, and its status is submitted. Let's go back while that's running and take a quick look at some of the information about this workflow. Again, if we want to check out the source code, you can see workflows are typically kind of this one file. It's taking a certain number of inputs and outputs. And the cool thing is workflows allow for like an optional output. So you can see there's a question mark there for our optional second read. And we were talking a little bit about, just before we started, about the environment that you run the workflow in. This particular workflow is running a tool called SeqTK, and it's sampling. And it's using an existing container that has that tool already installed. So I didn't have to install anything on AnVIL. I didn't have to run anything on command line to do that, but it's leveraging a tool that already exists. All right, and back to DocStore. And the nice thing too, you can see if the workflow has been updated lately, if it's been maintained. Something that's a little older may be a little less stable. And you can kind of see if there's any documentation down here. There's a little bit of tutorial in the, or it links to one of our tutorials that we've created. But there's also a little bit of documentation in the GitHub as well. Okay. And just like we were describing earlier, it inputs a FASTQ gunzipped or unzipped file and outputs a FASTQ file that's a subset of the original data. All right. And just like that, our job is done running. You can see that those workflows have succeeded. And let's go ahead and check out our data table once again to see what that looks like having run that workflow. Okay, so now I have a sample ID column. I have the full data in form of FASTQ GZ, and then I have a subsample of each of those set of reads that I could maybe use to do some debugging or some benchmarking or something like that. So let's just take a quick comparison of these files. I'm going to go ahead and check this out on Google Cloud. So this FASTQ file is pretty big, right? It's 11 megabytes. So, pretty sizable. It's going to take a little while to process if I were to run something on it. Not the hugest data we've ever seen, but it's nothing to scoff at. Versus if I were to go ahead and take a look at the subsample, go ahead and look at that on Google Cloud. This file is much smaller. Okay. So yeah, definitely a little easier to work with. And on many of these workflows, you can see there's supplemental files here to kind of check out, did everything run correctly? And this particular workflow doesn't create a ton of output, but you can see that it did a subsample with 1,000 lines. If you'd specified a different number, that would end up there, just to have a record of what you did. All right, so I'll head back to the slides now that that has finished running. Again, all this information is available in a self-guided tour. Basically, you can kind of rehash all the steps we just did, and how to set up the workflow, bring it into your workspace, and how to actually run it. And just a couple points before we wrap up and take some questions. We, of course, appreciate your feedback. I'll make sure that this link ends up in the chat. But yeah, we definitely want to hear what you think and how you're feeling about any of the things we talked about just now today. We also have a really awesome series that the team has created to learn AnVIL in 2 minutes. We know you're super busy, so definitely check those out to learn a little bit about AnVIL's capabilities really quickly. We also have our help.anvilproject.org forum where you can ask lots of questions, including about any of the stuff today. And AnVIL does so much stuff. And it is just like a huge team of awesome folks. But we know it can be hard to learn all of the things. So we hope you come back for another demo and learn something completely different from what we covered today. And just thanks to the rest of the team. It's a really awesome group of people I get to work with. So thanks.